All drugs bind to multiple sites on body tissues. We aim for the drug to interact with a specific receptor for which it has a high affinity as the result of the drug's ability to interact with a selective binding pocket in the receptor. When the drugs come into the proximity of tissues that express their receptors, they will bind to the receptors. In addition to these receptors, drugs also bind to nonspecific binding sites on tissues and in the blood. The definition of a nonspecific binding site is a site to which the drug binds with low affinity and which leads to no effect. These nonspecific sites can be components of tissues, such as proteins that circulate in the blood, proteins other than the receptor on the cell surfaces or inside the cells, or fat stored in various sites in the body. The number of these nonspecific binding sites is much greater than the number of receptors. The drug binding assay is used extensively in research and in drug development to determine where drugs bind, the affinity of their interactions with their receptors, and we can also use this assay to measure the number of receptors expressed by a cell or tissue. The basic components of a binding assay are a source of receptors, which can be a tissue homogenate from a human or animal source, or could be cells expressing a specific recombinant receptor, a buffer to keep everything at physiological pH, and a drug that's labeled in some way that we can easily detect. Typically, this is a radioisotope incorporated into the drug. To determine the binding of the drug to its receptors, we perform a saturation binding curve. A series of tubes are set up with a small amount of tissue in each, along with some buffer. The radio-labeled drug is then added in increasing amounts to the series of tubes, and they are incubated to allow the drugs to bind to the receptors. This usually takes about 30 minutes. After incubation, the content of each tube is rapidly filtered through a small filter that will trap the tissue with some of the radio-labeled drug bound to it, and also allow the drug that is not bound to the tissue to pass through the filter. After a rapid wash with cold buffer, the filters are taken and put into a machine that can detect the radioactive drug. Examining this curve shows that, as expected, increasing concentrations of free drug result in increasing amounts of drug binding to the tissue. However, we know that there are a relatively small number of receptors on the tissues, but the binding of the drug continues to increase as we increase drug concentrations well beyond where we would expect these receptors to have been saturated. Clearly, some of this binding is to components of the tissue other than receptors, and these components are the nonspecific binding sites. Therefore, it is necessary to determine how much of the drug is binding to receptors and how much of the drug is binding to the nonspecific binding sites. In order to do this, we must set up a parallel set of tubes, the same as the first set. In this second set of tubes, we add in an excess of the same drug, but which does not have a radioactive label. As in the first set of tubes, the binding that we measure in the second set of tubes is due to radioactivity labeled drug binding to the tissue. However, in this second set of tissues, the radioactively labeled drug can bind only to nonspecific sites. We know that the observed binding is to nonspecific binding sites because essentially all of the receptors on the tissue are bound by unlabeled drug, which we have added in vast excess of the labeled drug. This means that in the second set of tubes, the chances are that any given radioactively labeled drug molecule will bind to a receptor are very low compared to the chances that an unlabeled drug molecule will bind to that receptor. Essentially, the unlabeled drug has outcompeted the radio-labeled drug for all of the high-affinity binding sites, saturating the receptors with unlabeled drug, which is not detected by our machine. While the receptors will be saturated with unlabeled drug, there are many more nonspecific sites to which the labeled drug molecules can bind. Therefore, the radioactivity that we recover on the filter and measure in our second set of tubes will be due to nonspecific binding. To determine how much of the drug has bound to the receptors, we subtract the nonspecific binding from the total binding. We now see the curve flatten at higher concentrations of drug, since higher concentrations of drug are unable to produce any further increase in specific binding, which represents a saturation of the receptors at high concentrations. This allows us to estimate the total number of receptors in our assay from the maximal binding. We can also determine the affinity of the drug for the receptor as the concentration of drug required to occupy 50% of the receptors, which in this example is approximately 2 nanomolar.